Um, Hope y'all are having a good Friday. I know everybody's probably pretty tired, and just given by how many people are in here, I'm sure a lot of y'all that aren't here are probably going to be going to the game this afternoon. If you do go to the game this afternoon, please remember to not climb the Jumbotron, especially during a lightning delay. Let's not have some Pride students today. As always, just remember your quiz is due on Sunday. Nothing special. It's the same stuff you've been doing since the beginning of the semester. Again, remember that 2 of them get dropped, so it's not the end of the world if you have a bad one. I wouldn't use that as an excuse to just skip them, but it's not going to kill you if you miss one. Also, don't forget that the exam is Friday, November 5th. So that's 2 Fridays from today. So not this week, but not this coming week, but the week after. We'll do a review like we did last time. And just like last time, there'll be extra credit for it. Yes, it is a little bit more work, but given that you're going out and having to do observations anyways for that connected with biology assignment, I kind of expected that there should be some additional stuff on top of it to get extra credit, right? So it's nothing too crazy. If you have any questions about that, don't hesitate to reach out to me. Y'all know me. I'm fairly quick with getting back to y'all and try to answer as many questions as possible. So today we're going to be jumping back into what we had talked about previously with kind of talking about some of the mechanisms that drive evolution. Then we're going to keep on going down that path of genetic diversity and how that actually manifests and what does that actually mean for the scope of biodiversity on Earth. So right where we finished. I have no idea. I'm going to have to get a hold of SAS and tell them to send me something. That's the only thing I'm waiting on still from the previous tests. So I'm trying to get them to give it to me as soon as possible. I'm going to email them this afternoon because it's still not in my mailbox after 2 weeks, which is a little ridiculous, considering they had it to be the day after last time. So, but yeah, I'll make sure that if you took the test through SAS, I'll make sure that it gets taken care of. Don't worry about it. And if by some reason that they screw up and your test goes missing, we'll figure something out. Don't worry about it. All right. So where we kind of left off last time, we were talking about sexual selection as a driver for genetic diversity, driver for evolution in all these different populations. And like I mentioned, obviously building these complex nests, building these massive showy plumages like you see on peacocks, as well as building these massive horn structures and things for whether that be bighorn sheep to these very interesting rhinoceros beetles. All of that costs energy, right? And in the grand scheme of things, the only energy you really need to be using from a biological perspective is the energy to keep you alive and the energy to reproduce. So clearly, all that extra energy into these weird appendages is really necessary for that purpose. And that sexual selection can force traits that apparently reduce survival. Because at some point, say you're a large deer walking through the woods, if you have such a massive antler rack that it's getting caught up on all the branches and things, it's not exactly advantageous for you, right? But because that's what a female deer is looking for, you're more likely to be successful and continue reproducing. And it can actually cause some very interesting relationships where, say, for instance, we have active hunting in the States now for 250 years. There's been a selection pressure for removing individuals with large racks to mount up on a wall somewhere. Well, as a result, the animals that used to have all those nice, pretty genes for big, beautiful deer antlers are now gone from the population and aren't reproducing. And so over time, there's been a trend towards these smaller and smaller deer antlers. It's really cool. And how you can have all these different selections acting together to kind of push the population one way or the other. And the big thing here to keep in mind is that sexual selection is just another type of natural selection, and it's highly dependent on the species and the situation that species is in. There's some incredible relationships that exist here. Everything from there's lyrebirds out in Australia that have to have these bright blue colorations, and as a result, they steal like plastic and things. And so animals that are out in the more urban areas and more suburban are much more reproductively successful than males that are out in the middle of nowhere. And ultimately, you can see how something like that over time could shape these massive, beautiful plumages, the beautiful colorations you see on like African cichlids, that sort of stuff. And mutations can also cause evolution to occur. And remember, a mutation is just a simple change in the allele, right? You're just altering what's already present. Now, mutations on their own can't necessarily cause evolution to occur, because if it's a mutation that just manifests in one individual and that individual dies before it can reproduce, it doesn't matter. But if that mutation passes on either a neutral or some sort of beneficial allele, it's much more likely to survive and then reproduce into the next generation. But there's also something called genetic drift. Genetic drift is a true evolution per se. What it happens is basically, say for instance, you have a bag of M&Ms, right? And if you were just to randomly pick out, say 10 of them, and half of them were yellow and half of them were blue, and it totally happened to miss the green ones that were also in that bag of M&Ms, that's just pure happenstance, right? It's pure chance, just random. And as a result, we've lost that allele from the population, right? That's what happens with genetic drift. Ultimately, by random events in a population reproduction, it causes this change in allele frequency that would occur purely by chance. And this can be both detrimental and beneficial, just kind of depends. Now, yes, it does cause evolution to occur because the allele frequencies are changing within a population, but it's not like natural selection or sexual selection or anything like that, where you're being driven towards an ultimate goal. It's just sometimes random things happen. In the immortal words of Ian Malcolm, chaos, it's always out there. The flap of a butterfly wing can cause a hurricane in Taiwan. It's just all these little changes in a small system that's complex can lead to massive outgrowths and ultimate outcomes. And we kind of talked about this a little bit with some of the weird things that can happen with things like founder effects, which is where you have a few individuals migrating away from a population to start a new one. And because they're kind of subsection of that original population, they don't have all the genes present in that original population, right? They only have a small amount. And as a result, things that were relatively uncommon can become fixed in that new population. Say, for instance, you have some sort of homozygous recessive trait. In this case, I believe it's extra limbs or extra fingers and toes. Basically, what happens is that's normally part of life. It happens. There's a allele out there that does code for that, but it's homozygous recessive. You don't really see it in large populations. But for whatever reason, that allele became like 50% of a population just because of this founder effect where you subsetted out a small group of people that had a bunch of the same allele. And now you have an entire population where the majority, even though it's a homozygous recessive allele, have this six-finger trait. And there's another term for this called the population bottleneck. So in a founder effect, you're talking about organisms establishing a new population. Think invasive species. So things like the Cuban tree frog or the brown anoles that you see all around here, their genetic diversity is greatly diminished based off of them going from their original habitats over in Cuba and Haiti and places like that, then coming to Florida, because only a small portion of those individuals with a small portion of those genetics made it. Same thing here with population bottlenecks, just the mechanism's a little different. In population bottlenecks, you have a situation where some sort of artificial pressure, whether it be habitat loss, just random factors wiping out a particular area, say a crazy wildfire that hits a part of Australia where the species exists, and it wipes out an entire population, except for like 2 or 3 individuals. This happens all the time. And great examples of this occurs with the big cats, particularly the ones that occur here in Florida. In Florida, we have the Florida panther, right? Federally protected species. Well, in theory, at one point in time, the Florida panther was connected to the eastern panthers that ranged up through Georgia, South Carolina, what have you. So you had genetic movement going back and forth between all these different populations. Now, in today's modern age, because of habitat loss, loss of connectivity, and a bunch of other things, completely overhunting of most of the eastern population, all of the eastern cougars that are now known as the Florida cougars are isolated to a very small patch of land in South Florida. And as a result, their population numbers dwindled significantly. And through genetic drift, they lost a lot of that genetic diversity. And as a result, things that are deleterious started showing up. You wouldn't want to have sex with your cousin, right? But this was what was happening in these populations. It was as bad as some of them being as related as siblings constantly having to reproduce because that's the only mates they could find. And you get crazy, weird symptoms that started showing up in these populations where you had cougars that had really messed up tails that couldn't walk around very easily. They had, especially with the males, they had a lot of issues where the testes weren't descending so they could actually reproduce properly. A bunch of these issues. And the only way to fix it was to take a population of cougars that originally came from Texas, introduce a couple females, and do what we call a genetic rescue, where you introduce those new genetics, because in theory, these two species aren't really two species. They're kind of like closely related siblings. They're not really separate. And as a result, we were able to bolster those populations, put new genetics back into the Florida panther population, and they're doing a lot better. But that was an incredibly controversial call to make during like early 1990s, like the mid 2000s. So you can see where this stuff has a role and why it's important to keep track of these things. Now, like we've talked about, non-random mating causes evolution to occur all the time. Well, obviously, it's really obvious in something like sexual selection. There's a lot of other factors that influence mating, including geographical restrictions, physical access to the opposite sex, like we were talking about. When you only have access to your siblings and you don't know any better, that's what you're going to reproduce with. And as a result, you end up with really bad, messed up inbreeding problems. And so it becomes really important to understand this concept of gene flow, which is what moves alleles between populations, increasing genetic diversity over time. And this can change the allele frequencies in both populations. Because if you've got a rabbit that moves from this population to this one and it has two totally different alleles in each population, that allele is now present in the other population, and those population frequencies are going to change over time. This brings us back to the fact that evolution simply altered allele frequencies. And each mechanism of evolution is going to change those allele frequencies in different ways and in different strengths. And as a result, they're going to fall out of that condition for Hardy-Weinberg equilibrium and no longer be, eventually, these populations may push to become whole new species, subspecies, what have you. So let's talk about it, get away from it being a population level and talk about what this means for actually going from species to species and creating entire new families, entire new genera, that sort of thing. Now, usually when we think about evolution, it's on this much larger geological time scale. You know, ultimately, it takes a long time for these processes to happen. And while we can witness evolution in bacterial cells that are going to be reproducing every single day, an animal like an elephant or a tortoise that lives 100 years for a single generation, it's much, much, much harder to document the evolution of the time. And so it just takes more time. Now, ultimately, we use this geological timescale to divide the history of Earth into what we call eons and eras. So eons would be things like the total of the Jurassic, Cretaceous, and the Triassic period. Whereas era would be just the Triassic period or just the Jurassic period, sort of thing. Now, clues to evolution are pretty simple to find. Researchers can look at fossils from both the Earth, the actual geological structure of the Earth, body structure, and molecules to learn how these species are related to one another. Why do you think we know that dinosaurs are related to birds? It's because if we looked at these fossils over years and years and years and realized that there's a lot of similarities between things like a velociraptor, which has hollow bones, feathers, and all these sorts of things, and we're able to compare that to a modern day chicken. Plus, if you've ever seen something like an emu or a cassowary, it's not a doubt. They're dinosaurs. They will kill your ass. And ultimately, there's a huge mountain of evidence that supports this idea of common descent, where you started with one generic common ancestor, and over time, those branched off into new things and branched off to new things and branched off to new things. And think of it as kind of this classic view of a tree of life. You started with one basic bacterial cell, 1.8 to 1.2 billion years ago, and ultimately we ended up with humans. Not saying that we're the final form here, we're just one of the many, 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 many branches. And there could be something that comes out and replaces us someday. But there's still all these different things coming together and kind of extending from that single bacterial cell all the way down to us. Ultimately, the way to kind of look at this is through paleontology. You know, paleontology is the study of past life. It's not just dinosaurs. In fact, Florida has a really strong history with aquatic paleontology and mammal-based paleontology. Because during the ice ages, we were one of these very interesting places that had connections to South America. So we had some very odd critters showing up here. Whether it be giant ground sloths that stood 14 feet tall and had claws the size of most of your heads, to saber-toothed cats, Um, if you go back even further, you can see some things like Titanoboa, which was basically a 40-foot long anaconda. All of these things lived here in Central Florida. And if you really want a cool experience to see some of the more specific things to Florida, go to the museum in Gainesville. Really worth your time. It's really cool to check out. It's completely free for students, and use it while you can. And you can really see all these different things and how, looking back, starting 400 million years ago, which is the farthest fossils that we can date back here in Florida, all the way to now, how has the shape and the biology of Florida changed, even including humans that have colonized since about 100,000 years ago. Now, obviously, fossils are relatively plentiful, but they're not necessarily representative of everything. There are things that fossilize really well, and there's things that don't. And as a result, we really will probably never truly understand, just based off of fossils alone, what a population or a community would have looked like. We can have a pretty good idea. In fact, living here in Florida, we have some really awesome opportunities. There's a couple places kind of near Gainesville, as well as on the kind of, I think it's towards, what is that place called? The like Tomoka area, up kind of in between Daytona Beach and Jacksonville. There's a lot of these little rivers that are just full of fossils that you can like take a boat out for the day and pull hundreds of them in an hour, just from different mammal bones, shark teeth, stingray plates, which are the same thing as teeth, that kind of thing. What's really cool is we can use these fossils and basically place them along a timeline based off a wide variety of different factors, and use that to kind of record how evolution occurred over time. And again, these are anything from mammals, birds, reptiles, but you can also have fossils from bacterium and other microbes. It's exceedingly rare, but they are out there. And especially marine fossils in particular are extremely common here in the US, indicating this area might have been covered by an ocean. In fact, we know pretty well that Florida in particular was constantly either completely inundated with water or giant beaches, as far as the eye can see. In fact, how many of y'all have ever driven down the turnpike going from, say, Ocala to here? Have you ever noticed just how freaking hilly it is compared to the rest of Central Florida? Pay attention to it the next time you're driving through it. It'll be a lot more noticeable. It's because these were all giant beach dunes at one point. And over time, where the line for the actual beach and the waterfront has been pushed back to where it is now. And that's something to keep in mind too with things like modern day climate changes. Ultimately, Earth has always been changing. And we can probably just choose to adapt to it. We'll lose some things along the way, but it's not going to be as catastrophic. It's not going to be the end of the world. Earth always changes. Now, one of the things that we can look at is look at specific fossils called transition fossils, which are links to demonstrate these common ancestries. Probably the classic example of this is either Proto-Archaeopteryx or just Archaeopteryx. These are these very bird-like dinosaurs that have feathers, have claws, are starting to lose the teeth in their mouth and form more traditional beaks, that sort of thing. And you'll find that they have very similar features to what we see in birds, but again, they're that intermediary step. You often heard, especially in the 1800s, talk about the missing link between humans and other big primates. Most of those have actually, like the actual relationships have kind of died off over time, things like Neanderthals, Homo florensis, that sort of stuff. Granted, there was a bit of a racism angle in there too. That's really kind of nasty to talk about, but we need to mention it. But there was always this kind of looking for how do these things fit together? How do you transition from, whether you're talking about a human, from a chimpanzee to an adult human, to birds, from dinosaurs, and all those sorts of things. And like I mentioned, this fossil record is often incomplete. You're going to find that a complete collection is basically impossible. More often than not, any fossils that you do find are not going to have all the bones present, which makes analyzing them rather difficult, as well as the remains of extinct organisms may never fossilize. Fossils are often destroyed and some may not even be found yet. In fact, a kind of neat example of this is in the Badlands of South Dakota. You often have just these hills that erode away over time just due to wind and what happens. And during this time period, it slowly works away the side of these hillsides, and ultimately it'll expose things like Triceratops and T-Rex and that kind of stuff. Your classic big Cretaceous dinosaurs that everybody thinks about once they can see in a museum. But if those things are uncovered for more than a day or two, those fossils basically fall apart and just disappear. Like it was never there in the first place. So it becomes really important to find these things, get them into museums, get them prepared properly so you can actually look at them later on down the road. Another way that we can kind of start piecing some of these puzzles together is something called relative dating. And this is where we look at the rock layers that a particular fossil is found in and use that information to date how old that fossil is. Because yes, we talked about things like carbon-14 dating when we were talking about that first unit, but that only works for about the first 1.5 million years or so. And yes, there are other isotopes that we can look at, but in the grand scheme of things, when it comes down to it, it's a lot easier to look at the relative age based off of the rocks that it's all surrounding in, that compared to something like some sort of isotope-based method. And again, those methods have only existed for like the last 30, 40 years. So that kind of has always slowed things down a bit too. Again, like I mentioned, you can use absolute dating using chemistry. It's a more direct method to date the fossil, but it's a lot more costly. It's a lot more work intensive to do this. And depending on, you may just not have the ability to, depending on what kind of isotope you're dealing with. Now, like we've mentioned, it's pretty easy to do this with a lot of the Ice Age stuff because, again, that's probably only died off in the last 1 million years or so. So you can use carbon-14 pretty readily. Because ultimately, over time, carbon-14 is going to decay to carbon or nitrogen. And it takes about 6,000 years or so to do that, to go from 100% to 50%, another 6,000 years to go to 25%, and so on. It basically kind of acts like a curve that's approaching a limit where it can't actually reach the limit. You know, the limit doesn't exist. It goes away eventually to the point where you can't actually test it after some point, but it's what it is. Now, a big thing I do want to also point out too is scientists may not always have access to the fossils that are important, particularly in modern day stuff. And there's actually an entire show, I think either on History Channel or Discovery Channel, that is called Fossil Hunters, that kind of documented this whole deal. There was a T-Rex skeleton that sold for $31 million to a random private buyer overseas. That will never actually get looked at from a scientific perspective. And fortunately for this particular situation, turns out that the fossil was found on stolen land, and so it was able to be recovered and brought back to the US. But that's not always the case. In fact, the actor Nicolas Cage was involved in a very similar story where he bought a Chinese relative of the Tyrannosaurus from a Chinese fossil bed that had been smuggled out of the country. I don't know if he knew that or not, but he was just really into it for whatever reason, paid like $20 million for this damn thing. And because it was stolen, it was immediately confiscated and he almost went to jail over it. So it's kind of interesting how this stuff works out. So kind of an interesting question here. Which of these rock layers, A, B, or C, should have fossils with the most carbon-14 available stuff? Now remember, when it comes to relative dating, it's going to be based off of the stuff that's at the bottom. It's going to be the stuff that was deposited first. So usually, like, there's some really cool diagrams if you ever get to go to places like the Grand Canyon or Providence Canyon up at Georgia, that sort of thing, where you can actually see where they mark out when they think that those different layers were laid down. And you can look at it and say, oh, this was probably 400 million years ago. This is probably 200 million years ago. This is probably within the last 1 million years or so. So based off of what we talked about with carbon-14 dating, which one of these makes the most sense for going to have the most likelihood of using carbon-14? Hey, yeah. And that's simply just because carbon-14 only works for a certain period of time before it just kind of, there's not enough carbon to detect. So here's another interesting question. Researchers use radioactive isotopes with a 25,000-year half-life to date a fossil that's 100,000 years old. This fossil contains blank as much isotope as it did as a living organism. This is relatively simple math. Anybody want to take a shot at it? Yeah, exactly. It's 1/16. And the simplest way to do that is 1/2 to the 4th. Pretty straightforward. Now, obviously, we've been talking about how all these fossils and stuff fit together, but we often need to incorporate what we see in the earth as well, looking at both the geochemistry that involves with all that, as well as just the general geology of the earth. As that leads us to something called biogeography, which considers a species' geographical locations in relation to things like mountains, oceans, all that sort of stuff, which can influence evolution. Now, plate tectonics is a relatively recent phenomenon. It was only really confirmed in the last 50, 60 years, which is rather surprising if you think about it. But ultimately, we know now that at some point in the Earth was one giant supercontinent about 250 million years ago. And over time, it split up into all the continents that we have now. Now, at some point down the line, in the next 200 million years or so, more than likely, it's all going to come back right together. And that's all based off of these convection currents that occur in the Earth's core and mantle that move these crusts of landmass around on the Earth. In fact, this is where earthquakes come from, this is where volcanoes come from, all that sort of thing. Now, when these continents move around, that's going to shape evolution. Say, for instance, if you're South America or Australia, that got isolated from all the other continents about 50 million years ago, yes, you have mammals, but the mammals that you have are the most basal form that evolved in a totally different direction than the ones that we have in other places. Think about Australia, right? What is Australia most known for? Yeah, things like kangaroos, koalas, wombats, platypus, all this kind of stuff. Relatively speaking, they're very basal level mammals. Things like the echidna and the platypus are the only mammals that we know of that still lay eggs. And especially when it comes to the other marsupials, things like the kangaroo, the wombat, the koala, all that stuff, they don't even have a full placenta. They can't go to full term like we see in other mammals. And as a result, their offspring are born very, very tiny and have to go through that pouch development. Where's the other place that you see marsupials? There's one right here in America, but it came originally from another continent. You see them around all the time. Possums, yeah. All throughout Central and South America, there's about another 10 to 15 other species of possum, again, another marsupial. And the reason for that is at one point, Australia and South America broke off by themselves. They kept all the marsupials while everything else kind of developed and ultimately outcompeted marsupials. And as a result, Australia developed its own wildlife, South America developed its own wildlife, and when it merged into North America almost 1.5 million, 2 million years ago, yeah, we have the North American possum, but that's the only thing that's been successful and has moved up. For the most part, the animals have moved down. The things like the jaguar, mountain lions, all that stuff that existed in North America moved down into South America. This is called the Great Columbian Exchange. It's really fascinating stuff. And what's really interesting now is particularly in Australia, they have a very hard time with invasive species, particularly mammals. Because for whatever reason, rats, rabbits, dingoes, a lot of that stuff, which dingoes are arguably just another invasive species. It's just, they've got 100,000 years on most of the other ones. Those all didn't exist on Australia prior to about 100,000 years ago. And as a result, you have these massive imbalances in how these animals are able to outcompete with each other. And things like wombats, quolls, a lot of your more distinct classic Australian wildlife are going extinct at massive rates. You see this pretty consistently with island stuff, but it's even worse on a place like Australia because it's so massive and you have so many opportunities for things to have evolved over time, and now they're just being wiped out. And again, this all comes back to that the species distributions can reveal these evolutionary events where South America might have broken off from Africa and Antarctica and Australia, and at some point they came back together between Australia and South Africa, or South America, and all this kind of fun stuff. And ultimately, we can use this information to shed light on these evolutionary events. For instance, we talked a little bit about Alfred Wallace and how he was able to kind of independently derive the concept of evolution around the same time that Darwin did. And a lot of that had to do with something called Wallace's Line, which is a spot somewhere in between Indonesia and Papua New Guinea. That basically, at a certain point, The species that come from this side are more like the stuff that shows up in Australia. So you get a lot more of the pythons and the marsupials that you normally associate with Australia, versus coming down from the Malay Peninsula, you get things like rhinos, bears, tigers, what have you, elephants. So while we can talk about biogeography, there's another thing that kind of reveals these relationships between one animal to another. And we can look at the anatomy to do this. And this is relatively easy to see. The one term I do want to point out to you is something called homologous structures, which are structures that are inherited from a common ancestor. In other words, humans have the same arm bone structure as lions, bats, seals, and all of that is because at some point all mammals were based off of the same damn body plan. And here's a really great example of this. Here you have 4 different species. You have humans, domestic cats, probably a bottlenose dolphin, and a bat. All of these have the exact same bones in the exact same order. The only difference is the length of them and how they're structured. So are they thicker, are they thinner? Where do they get exactly placed in the arm bone, that sort of thing. So if you ever look at, like, say, a dolphin skeleton, they look kind of like a creepy, washed up mermaid. Because in some of them, especially the baleen whales, still have hip bones, even though they've long lost those rear legs over time. Now, this homology is going to demonstrate these evolutionary relationships. Because all these things have homologous structures that have developed differently over time, yes, they are slightly different, but you can still see how they came from one another. And this is all based off of that differential selective pressure that pushes one species to change to fit the environment that it's currently occupying. Like I mentioned a couple seconds ago about the whole hip bone thing, a lot of animals, because it's just not advantageous to completely get rid of something, have what we call vestigial structures. These are leftover things from evolution that have no purpose. And again, this is one of the reasons why I talk about evolution is not perfect. You're not aiming for a perfect overarching goal. It's just kind of happenstance happening over and over and over again. So here in this vestigial structures, you can get things like in moles, even though they don't have a use whatsoever, they still have eyes. They're basically just a little patch that sees light and dark, and that's it at this point, but they're still there. They're not really useful, but they in things like boas and pythons, you can often see a little nub that occurs right back near the cloaca. That's what's left over from their actual legs, their hip bones. It's really cool. And it's one of the ways you can actually definitively say that pythons and boas are more recently derived, or are something that came very early on in the development of snakes compared to stuff we have around here. Yes?
Would the appendix be considered a vestigial structure?
It's been argued that for sure. She asks, would the appendix in humans be considered a vestigial structure? I would argue that it is, just because for the most part, it has no real purpose. Although, honestly, as scientists, we're constantly discovering new things every single day, even about humans that we know probably the most about compared to all other organisms. So I'm not willing to quite just write it off and say, yeah, it has no purpose, but I think it probably is vestigial at this point. And of course, like we've already mentioned, at some point there was a whale ancestor that had 4 limbs. And if you look at modern day baleen whales, they still have that vestigial hind limb that's still tucked up inside of their body. And what's really cool is now you can look back at the fossil record and see how developed those hind limbs are, and use that as a way to gauge how far along in development were you from these more crocodile mammals that are crocodiles versus what we now consider modern day whales. But don't get too hung up on this stuff, because there are sometimes where structures that serve very similar purposes were evolved independently. We call those analogous structures. For instance, that cute little mud puppy and that crayfish both have gills. They're both designed to do the exact same thing, but they didn't evolve together, right? It's not because, you know, salamanders are closely related to these crayfish that they have the same gill structures. It's just because they've happened to follow and fit into these environments. And this is especially important when you're looking at organisms that, in particular, are evolving to fit a very particular niche. So, for instance, if you want to talk about analogous structures, probably a good example of this would be something like comparing a pterodactyl to a bat. both have these very long, leathery wings that are made off of the structures of their forearms. But they're done differently, and as a result, they're considered analogous structures, and it's not because they came from a common ancestor, they evolved independently. Another great example of this is something like snakes, legless lizards, and some of the salamanders that have completely lost their legs. All three of these animals have decided that it's better just to be living underground or fossorial and ditch the legs because they just get in the way, and so they've evolved without them. They're analogous because they all evolved independently. It's not like lizards just all of a sudden said only this branch can lose their legs. There are multiple branches in lizards where you see loss of limbs at some point. And again, these are the result of convergent evolution, where you have animals that have lost things or evolved particular traits because of the habitats they're evolving to fit into, not necessarily because they're coming from the same common ancestor. So here's another example. When you're looking at the streamlined bodies of dolphins and sharks, this is something we call fusiform, which is torpedo shaped. These bodies, plans of the animals are either homologous, vestigial, or analogous. Which one are they? They're analogous, right? Because it's not like sharks and dolphins both evolved to the exact same form at the exact same time from the same ancestor. Sharks evolved hundreds of millions of years before dolphins ever were thought. So that's why it would be considered analogous. So as a result, it's a product of convergent evolution, which would make it both C and D. Now, one of the ways we can see these patterns is through embryonic development. Embryology in particular, you can actually watch how pretty much all vertebrates have almost the exact same looking structures through about the first 16 weeks of life. If you were to do an ultrasound on a human and compare that to a tadpole developing openly in an egg, they look almost exactly the same for like the first couple bits of life. It's really fascinating. And ultimately, how those things change over time since then is what changes ultimately into their adult form or their juvenile form, however you want to put it. So again, these anatomical similarities are often most obvious in embryos. And notice how much more similar humans and chimpanzee skulls are in their structure and their features compared to the adults. Has that very like Alien from Alien Resurrection look to it, where it's kind of this big bulbousy looking head. But over time, chimpanzees are going to grow those much stronger jaw muscles and a smaller brain casing. And again, that's all for stronger actual jaw muscles, which allow them to break down things a lot easier than humans can. Although humans have a nasty bite too. But still, you can see how like these things can change over time. And like I was mentioning, the early vertebrate embryos appear very, very similar, whether it be a chicken, a mouse, or a human. Granted, the timing does kind of differ depending on the level of development and all that sort of thing. But in humans, it's probably about what you're going to see through the first 10 to 16 weeks, and it's going to be very, very similar to what you see in very early stages of development for a lot of other organisms. Now, there's something called homeotic genes, and these are going to be the genes that actually control this embryonic development. So these genes are needed for body structures to develop normally, and as a result, small differences in gene expression can make massive differences between a limbed and a limbless organism. For instance, when you're comparing a classic chicken or like a python, there's that slight variation in that gene, which is more about timing and production of that protein in a very particular time period, results in a complete body plan that just no longer has limbs whatsoever. Now, mutations can occur in these homeotic genes to produce dramatic body structure changes. For instance, you see this a lot, and particularly with fruit flies, just because fruit flies are just really easy to reproduce, really easy to have massive numbers of generations quickly. And as a result, humans have used them for kind of a guinea pig for a lot of these evolutionary concepts. And just basic changes in these things can result in legs growing where the antenna should be, or malformation in the wings, so they're no longer flighted animals. They have to basically walk around. And we can even use these things to our advantage. For instance, I used to keep dart frogs, and if you didn't want your entire house covered in fruit flies, having them flying all over the place, you'd buy the flightless ones, which is just simply a small change where it cripples the wing development, and as a result, they're nice and easy to feed my dart frogs. we're kind of running a little bit low on here in time, so I'm just going to skip this. Now, we've talked about all these anatomical features and geological features, but another way we can look at and determine if something is related or not is just by looking at the genetics. So we can look at the molecular patterning that you see in the DNA and thus compare the DNA sequences and their protein sequences to determine evolutionary relationships to an unprecedented detail. This allows us to do what we can never do with just anatomy alone. And as a result, a lot of places, particularly when you're describing new species, require almost a combination of approaches where you need to at least have some genetic material and be able to analyze those genetics, as well as the anatomy aspects of things to be able to compare and say this is definitively a new species. This gets kind of complicated very quickly because things like salamanders, from a genetic perspective, they may have differentiated 5 million years ago and be completely isolated, even though they look almost relatively the same because you have this convergent evolution factor keeping them in a particular body plan. And as a result, one of the salamanders that I'll bring in here at some point, there are 8 different species of this thing. They all look exactly the same, but they differ so much genetically, it's not even funny. And so as a result, we've had to split them out. Now, it does kind of get somewhat frustrating because over time we realize that some genetic techniques are better than others. And we realize that, oh shit, we probably shouldn't be just looking at mitochondrial DNA. Why would you not want to just look at mitochondrial DNA? Exactly. It's only maternally inherited. So you're totally missing a whole aspect of genetic diversity, but you're only looking at something and it may cut your pool and say, oh, these look a lot more differentiated than they actually are. And so we've had to go back to incorporating both mitochondrial and genetic, like what we call whole genome DNA as well. So ultimately, we can use these sequence similarities through inherited common ancestors to kind of depict where exactly on the tree of life all these things fit together. It's not very easy. And oftentimes you have to get away from just using a couple genes and go towards what we call genomics, where we're using the entire genome of the animal, all 2+ billion base pairs of an organism to differentiate this from another thing. And that becomes really complicated really quickly, because obviously there's going to be just some natural variation that occurs in a population, right? You don't want to just totally skip that and just say everything is the same thing. Plus, you need to keep in mind too that organisms are always perpetually evolving, right? That's what we just spent a whole day talking about. So it's important to understand that there may be populations that are differentiating before your very eyes. And so it's important to catch these kinds of things, incorporate that into your study designs. Now, a big thing to keep in mind too, though, is that DNA changes are common for many organisms. Cells add new functions by acquiring DNA from viruses and other organisms. They duplicate their genes over time, changing gene expression or by mutations. All these different things factor together to slowly change the genome over time to where organism A is no longer related to organism B. And it gets us away from this whole, I'm sure you've probably heard of something called the biological species concept that says in order for it to be a true species, it can't reproduce. That's bullshit. There's some fascinating hybrids out there that are completely able to function. Things like lions and tigers reproducing together. We're pretty damn sure that a lion and a tiger are separate species at this point, right? They can still reproduce, they can still have hybrids, and those hybrids can often reproduce themselves. So the biological species concept doesn't work. And so we use a lot more of the genetic underpinning of that to kind of inform how we think of things now. Ultimately, this DNA changes how evolution works. And ultimately, remember back to evolution is just simply those alleles changing frequency. And so if you have new alleles being introduced into a population, that changes how evolution is going to occur in a population over time. And you can use things like protein sequences that are homologous. Think about our homologous structures in animals. You can use very similar kinds of approaches to think about a particular protein in an organism. We can use that to basically say, based off of these very specific protein sequences, we can compare the differences between something like a human to all these other creatures. So for instance, here in this cytochrome C mitochondrial protein, there's 0 differences between chimpanzees. There's 1 difference between rhesus monkeys, 10 differences between cows, 18 differences between bullfrogs, 25 compared to fruit flies, And finally, 40 compared to yeast. But they still code for the same damn protein. It's just slightly different. So cool. What's really neat is based off of the number of changes, we can do applies to something called the molecular clock, where we incorporate fossils as well as these anticipated mutation rates, because we know relatively how often mutation occurs, especially in particular alleles and genes. We can combine all that information together and say, based off of the amount of changes and where those changes occurred and how long ago they were, you can basically say, okay, here's when this organism broke off from this organism, and actually date it to a specific time period. And what's really cool is oftentimes these are pretty damn close to the actual fossils that we find later on. So, say for instance, mutations in a gene occur at a rate of 1 nucleotide every 10 million years. There's a gene sequence that differs by 6 nucleotides between 2 related organisms. How long ago did these organisms split off? Pretty straightforward, right? You know that there's 6 nucleotides difference, and it takes 10 million years for one of these differences to occur. You can roughly approximate that the answer would be, somebody say it? Hey, yeah, 60 million. That's wrong. Ignore that. Sorry, the book screws me on some of this shit. It really pisses me off sometimes. But what's really cool is we can also look at, it's not, it's definitely 60 million years. Ignore that. So one of the classic examples of this, and a great example when you're trying to compare evolution, are snakes, especially in regards to their very similar cousins lizards, right? Ultimately, all snakes are lizards, but not all snakes. Yeah, vice versa. Basically, long story short here is at some point a lizard ancestor evolved into snakes. They lost all their limbs and all that sort of stuff. But limblessness isn't only in that snake branch. It's in a lot of other different things. In fact, we have glass lizards here in Florida, but they're still lizards. They still have eyelids. They still have a lot of traits that make them very specifically a lizard. But they look just like snakes to the untrained eye. If you're looking for them, you're going to think it's just a snake. And so you have to incorporate all these different things together. And here you can actually see a really cool phylogeny of all these things. Everything from Acrodonta, which are things that your true chameleons that you associate with Madagascar and Africa and parts of Australia. Iguanids, beaded lizards, which are your venomous lizards from Central and South America, things like your Gila monsters and what have you. But what's really cool is we can look down here and see that serpents broke off of this group here that includes things like iguanids and helodermids and a bunch of pyranids, which are things like your Nile monitors, which are now invasive all throughout South Florida, as well as Komodo dragons, which are also venomous. It makes sense all of a sudden why serpents, things like vipers, cobras, what have you, may be able to develop venom because it's evolved multiple times on that branch compared to everything else. Still really fascinating. All right. So as always, remember your quiz is due at its normal time. Please let me know if you have any issues or anything. I'm always happy to help you. Again, don't forget the quiz. Don't forget the connecting with biology, and don't forget the extra credit assignment. They're really not that hard to do. If you're looking for a place to go knock out a lot of your observations really quickly, Apopka Wildlife Drive is awesome. Definitely worth the time to go out there and just drive around. You can see at least 20 gators, about as many birds as you can possibly need to see. The Kiowa Springs is really beautiful. It's a great place to go check out. You have the arboretum here on campus, but you're going to need to go out at specific times to make sure you can see the birds and that kind of thing. Yeah, there's plenty of places to go out and different things. Right around here too. Yeah.
Yeah, no, I saw it.